All right, hi everyone. My name is Jens Puschhof. I'm a PhD candidate with Hans Klevers in Utrecht. I want to talk today about organoids, so 3D tissue culture systems, and in particular recent developments, 4 stories which we have published last year, with a particular focus on organoids of the snake venom gland and how we studied bacterial contributions to colorectal cancer using the organoids. At the end of my talk, I want to give a one-slide sneak peek into 2 other projects, one on the novel coronavirus and how we can model infection with this virus with organoids, and the rare intestinal cell types, and also how organoids help us understanding these things. With my very first slide, I want to give an intro on organoids. So what I'm talking about are adult stem cell-derived organoids, so we can grow them from patient biopsies or from animal biopsies. We can expand them indefinitely in culture. They're genetically stable, so you don't need any transformation step to grow them. They grow in 3D. We embed them in 3-dimensional matrix, matrigel, or basement membrane extract to grow. And they are heterogeneous. They contain both the stem cell, but also the differentiated cell type of the particular organ they are derived from. With adult stem cell-derived organoids, we grow only the epithelium, but not the mesenchyme. So they're a reductionist model system, but can be very useful to study exactly these epithelial processes. And finally, as mentioned, a single adult stem cell can be enough to grow an entire organoid model. In the first story I want to talk about, we grew the first reptilian organoid system from the snake venom gland. And this has been a collaboration with Jorik Post and Hugh Böhmer in the lab, but also a large group of collaborators worldwide to get all these snakes and understand what's happening in these organoids. We started out with 4 main motivations to do this. First, we wanted to learn something about stem cell biology because organoids were first derived in 2009, and in the last 10 years we've learned a lot about how adult stem cells work in mammals. But if these principles would be conserved also in a reptile, that was not clear. So first of all, we wanted to understand how conserved adult stem cell biology is beyond mammals. Second, venom production itself is a fascinating process, and it's pretty hard to study for you cannot just CRISPR a snake and they have longer generation time than mice. So it would be really useful to get organoids from snakes to be able to study these processes of venom production and secretion in more detail. Third, snakebite is a major global health problem. There's more than 100,000 deaths each year based on snakebite, and the antivenom production still largely relies on having large amounts of the venom itself. So we thought if we can standardize this process, we could help to make a next generation of antivenoms based on organoids. And finally, the toxins in the venom are also quite biologically active substances, and there are 6 FDA-approved drugs based on snake venom. So we thought probably that's a good one to produce the venom biotechnologically with the organoids and see if we can use them as a production platform for these bioactive molecules. With all this motivation in mind, we set out to derive the first organoids, and the process was surprisingly easy. What you see up here is the protocol, which is very similar to what we do for mammals. Here we often started with an egg with late-stage embryos, dissected out the venom gland, dissociated it to small chunks of tissue, so probably 30 cells apiece, and then we embedded them in this 3-dimensional scaffold, and they started growing into cystic organoids, looking very similar to what we get from most mammalian organs. The main difference is that they grow at 32 degrees, more closely recapitulating the body temperature of a snake. Very interestingly, as a first finding, we saw that the growth factors required to support the growth of these adult stem cells to organoids are very similar to what we know from mouse and human, and these are even mouse and human growth factors, so they are conserved enough to support the growth of snake stem cells, just highlighting how conserved these key stem cell pathways are. When we published the paper last year, we had 9 species established, and you see them all here, very colorful snakes, but the organoids look reasonably similar under the bright field microscope. So to give you a better overview of what they really look like, I have a short video here on the fluorescence microscopy. And we see as we fly through one of these organoids that they're a monolayered epithelium. They're quite polarized with an actin layer towards the middle, shown in blue here and just coming back up. And we even can spot some dividing cells up there, for example, in the organoid. To really understand what's going on in the organoids, we went one step further and performed transmission electron microscopy. And this was the first moment we started thinking that they could really produce venom, because when you look at this, you see a monolayer epithelium, a nucleus towards the basolateral side, but you also see a lot of vesicles towards the apical side of these cells filled with gray or even black substance. And when you compare this to an image of a real venom gland, you see quite a striking resemblance of these vesicles which are known to be filled with venom in the original gland. Interestingly, we also see heterogeneity here. So one organoid differentiated very well, and you see dark vesicles here at the bottom and a dark filling of the organoid, whereas the organoid on the top of the screen did not differentiate as well, and you see that there's no dark substance inside. So we're wondering, do these organoids keep accumulating venom in the middle? And to address the question, we teamed up with Jeroen Kool and Julian Slagboom in Amsterdam to characterize what's in there. So we lysed the organoids, we got whatever was in their lumen, and then they ran LCMS to characterize the protein profile in there. And indeed, when we compare the real snake venom on the top and the organoid supernatum at the bottom, we see a lot of traces which have an extremely high correspondence in size. And when our collaborators ran tryptic digest to characterize what is produced by those, what are these peaks, we see that these are mostly belonging to the family of three-finger toxins, and that's a class of neurotoxins from the snakes. All of these data here are from Aspidolaps lubricus, which is the Cape Coral snake and our poster child snake for this project, basically. These three-finger toxins are most well known for blocking the action of acetylcholine receptors. And so our next question was, now that we know that these organoids make venom, is it also functional? And to check that, we teamed up with Mimetas, a biotech company in Leiden in the Netherlands. And they have so-called myotubes on the organoplate. So these are mouse muscle cell line cultures grown in these fibers, which express acetylcholine receptors. And when exposed to a green calcium dye, you do see that waves of calcium signal are propagating through these myotubes. And the video down here is already playing, so the toxin exposed ones, where we took the organoid venom and we exposed the myotubes with them, and we see that these green signals are gone. We can also quantify this and see that indeed there's a lot of calcium wave propagation in the control exposed myotubes at the top, and this signal is almost gone with the toxin exposed cultures. And the quantification looks like this, and serving as evidence that the organoid supernatant is enough to block the firing of myotubes, which is very similar to a snakebite paralyzing muscles of a victim. Great work again by Mimetas, and please Nike Weber, Sander Spijkers, and Thomas Oliver here. With that, we had one remaining big question, and this is the cellular heterogeneity of the venom gland. It's not really clear if there's one cell type making all the different toxins which make up venom, because venom is a mixture of dozens of different compounds, or if there are specialized cell types contributing the different toxins. To address this, we performed single-cell RNA sequencing on our organoids and primary venom gland tissue. So this is a T/E map from single-cell RNA sequencing data. Every dot here is one cell, clustering by similarity. We have two different media. In yellow, you see expansion medium, so where we enrich for stem cells, and in black, we have differentiation medium, where we push them towards the venom production, what we know from our experiments before. And when we look at where most toxins are expressed, they really cluster in the lower half, so in the differentiated cells, which makes a lot of sense. But now our question was, do different cells make different toxins? And indeed, we see that there's a population down here, which predominantly expresses three-finger toxins, so the neurotoxins we already were talking about earlier. And interestingly also cofactors for the folding of these, like protein disulfide isomerase here, extremely well corresponding with the expression in each cell. But for other classes of toxins, like the C-type lectins, we find different populations, like marked in purple here on the left, for example. And for other toxins, yet again, we have more populations expressing these. So this is the first evidence that we could really have different subpopulations of venom gland cells making different toxins. What we did to sum this up was to use RNA scopes, so fluorescent in situ hybridizations, to check where this expression is located, because we don't have the spatial information in the single-cell RNA sequencing. And when we look, for example, at this cofactor, the peptide disulfide isomerase, we do see that it is located in the glandular structures, but not so much in the stroma or the duct of the venom gland, just adding some spatial information to the patterns we observed in our organoid single-cell RNA-seq. To sum up this project, we do have 3 take-home messages. First, organoids are grown from adult stem cell, and we can grow adult stem cell-derived organoids, not just from mammals, but also from reptiles. So, of course, now we and others are pursuing many different species to see how far we can stretch this organoid technology beyond the traditional mouse and human models. Second, the venom gland organoids of the snake produce functional toxins, which we could demonstrate with some chip assays. And finally, we have some evidence of heterogeneity in the venom gland, of cellular heterogeneity, meaning that different cell types could be producing different toxins, which is a great basic science insight on how this organ is organized. With that, I want to move on to the next project, where the only 2 unifying factors are that we're using organoids to make a discovery, and that we're talking about toxins in one way or the other. In this project, we set out to check if bacteria can contribute mutations to colorectal cancer development. Just like the last one, it's a 3 PhD student collaboration with Caetano Pleguezuelas-Manzano and Axel Rosendahl-Huber. As an introduction, you may all be aware of that our gut harbors a lot of bacteria, many different species, some of which have beneficial effects for our health, but others can also contribute to the development of diseases. And one of those is called genotoxic E. coli. Genotoxic E. coli is pretty much a normal E. coli, just it does have an additional gene island, the so-called P.A.C.E. island. And on this P.A.C.E. island, it encodes a set of enzymes which produce the genotoxin colibactin. So P.A.C.E. positive E. coli produce the genotoxic compound colibactin, and that's what we study in this project. We started to answer one pretty fundamental question, and this is, can bacteria contribute specific mutations in human cancer? And because colibactin is well known, these bacteria are well characterized, we chose these strains to address the question. To check if specific mutations are introduced, we used another method, and this is called mutational signature analysis, I think published first in 2013. It looks at the context of where single mutations happen. So here you see the examples of single base substitution, where one nucleotide gets replaced by one other, shown in the middle here, like a C being replaced by an A. And we'll also take into account the base 1 upstream and the base 1 downstream of the mutation. So when we go through all of these combinations, we end up with 96 combinations. And now we can go through the genome of a cancer patient and count how often does each of these 96 combinations happen. And when we do this, we plot all the 96 combinations on the x-axis, and we count the mutations per combination. We get a profile or mutational signature, and this has been found to be very characteristic for many oncogenes or for many carcinogens, whatever you can think of. So this, for example, is an example of UV irradiation and its mutational footprint, where you see that C2T mutations are enriched, and in particular in some special trinucleotide contexts. These have been found by now for a lot of different carcinogenic factors, but they hadn't been described for a bacterium. So we're looking for one of these mutational signatures caused by genotoxic E. coli. To do this, we used a wild-type genotoxic E. coli strain and an isogenic knockout, where one of these enzymes on the P.A.C.E. island is removed, so they no longer secrete functional colibactin, serving as our negative control for anything colibactin specific. So, where do organoids come in? This is now, they are wild-type epithelium, as mentioned in the beginning, so we really start with a very clean genotype. We can grow them clonally from a single cell, so they all start with the exact same genome. And now we can expose them to bacteria. Because organoids are polarized and their luminal site is also on the inside of an organoid, we have to inject the bacteria to make it as physiological as possible. And this is what we did. And to allow them to accumulate mutations, we did it over a time frame of 3 months. every 2 weeks injecting bacteria, some days later removing them with antibiotics, splitting the organoids, and then repeating the cycle. And what we're expecting or hoping for was that in the wild-type E. coli exposed organoids, mutations would accumulate, shown on the left here. You can imagine that every single cell will capture different mutations, so sequencing whole organoids may be very tricky to analyze in the end. So there's another advantage of organoids, that we can grow them from single cells. So we can dissociate those and, in a second step, grow clonal organoids. So this is what we did, associating the single cells, growing clonal organoid lines. And once we grow these clonal organoid lines, we can isolate their DNA. They will all have the same genotype now and capture just mutations which were caused in one cell throughout this co-culture. And in the whole genome sequencing, then we're hoping to find both unspecific mutations just through normal replication errors, but also some specific mutations in the wild-type E. coli exposed organoids. So when we looked at all these single base substitutions with mutational signature analysis, we found the following things. On the one hand, in both the control bacteria and wild-type bacteria exposed organoids, we find a pretty broad background signature, which was expected, but only in the wild-type E. coli exposed organoids, we find another signature. And because all of this was done in parallel, we can now subtract these background mutations from the wild-type E. coli induced mutations, and by that come up with a pretty clean signature, which we call SBS-pks. And this is for single base substitutions caused by P.A.C.E. coli. And when we look at what kind of mutations are characterizing this signature, we see that everything here is a T to N mutation, so a thymidine getting replaced by something else, and it's all in the context of neighboring adenosines and thymidines. So definitely something about adenosine thymidines going on with that signature. These have all been animations if you haven't noticed, so I'm going to show the real data now, and you see that it's just the same. This here is the wild-type E. coli induced signature, the background signature, and the subtraction-derived SBS-pks. We can do this not just for single base substitutions, but also for insertions and deletions. And without going into too much detail here, when we look at insertions and deletions, we find a strong enrichment of this class, which is the deletion of a single thymidine in a poly-T stretch. And I'll come to that later. Trying to understand what's going on a little bit better, we looked further than just one base upstream and downstream. And this was really surprising, this project, that it's not the thymidine or adenosine one base upstream or downstream in the green bar here, which is most telling of the E. coli signature, but it's really 3 bases upstream that we have a striking enrichment of an adenosine on the same strand as the thymidine, which gets mutated. While we were pursuing this project, there were some great papers being published in Science on the chemistry of colibactin, showing that it can bind to adenosine and cross-link to adenosines in the DNA. Now, taking these pieces of information together, we think the following thing may be happening. And this is that colibactin is binding to the adenosines on the paired strands of DNA in about 3 bases distance. So it's cross-linking adenosines in 2 strands, and during the repair process, something goes wrong, and we have a thymidine in the end being replaced, which of course also means that on the paired strand, an adenosine will be replaced. All of this is just circumstantial evidence from these studies, and I think this is really the next exciting step to understand this mechanism better, based on the characteristic mutational patterns we found. We found a very similar pattern for the indel signature, and this is that our poly-T stretch here has a poly-A stretch directly adjacent to it. So also here we think it may be colibactin binding to 2 paired adenosines, but why exactly one of these gets resolved with the deletion of a single T and the other one by the replacement of the T is still pretty much unclear. So much for the molecular part and for the organoid part, and of course as an organoid lab we can get very excited about this, but in the end we have to show that this is happening in patients to be relevant. And this is what we tried in the second part of this project. Now that we have identified 2 mutational signatures in organoids, seeing if they also happen in colorectal cancer patients. And this we could indeed show. So when you look here, we have SBS-pks, so our single base substitution signature, and our indel signature. And what we did was analyze a cohort of more than 3,500 whole genome sequences of metastatic cancer of various different primary sources. And we asked the question, what percentage of their mutations is best explained by our new signature compared to all the other signatures? And when we do this, we see that only a small subpopulation of all these 3,500 metastases has a large number of mutations caused by SBS-pks and ID-pks. And when you look at the color, almost all of them are orange, meaning that they're coming from colorectal primary cancer, which is, I think, a great finding. We start with a gut bacterium injected to organoids, identify these signatures, and when we look for them in patients, again, we find them back in the colon and rectum, showing that most likely they're indeed caused by a gut bacterium. You also see a lot of gray dots here, which are all the different cancer types, so they don't have a large contribution, and an occasional head and neck and urinary tract cancer with a high contribution of these signatures. And these are both organs which get infected by E. coli, but which have not been implicated with P.A.C.E. coli as much. So we think this could be a great starting point to start looking into other cancers beyond the colon for these mutational signatures and for contributions of P.A.C.E. coli. More than just the presence of random mutations, we could identify a number of patients with potential driver mutations caused by SBS-pks and ID-pks. For example, these are patients with the mutation A.P.C.E, so one of the most prominent colorectal cancer driver genes, and every red dot here is one patient with a premature truncation in A.P.C.E caused by SBS-pks or ID-pks, meaning that indeed these bacteria could be contributing to driver gene mutations. And this is something which we're also investigating right now, to what extent they play a functional role in the early stages of colorectal cancer initiation. To summarize this part, we set out to look for a first mutational signature caused by bacteria in human cancer. And using an organoid experiment with a long-term exposure to these bacteria, we identified two mutational signatures, SBS-pks and ID-pks. When we look for these mutational signatures now in patients, we find that they're enriched in actually two large cohorts of colorectal cancer by now, and we're talking about 7% of patients who have high levels of these signatures. So it's a reasonably large proportion of colorectal cancer with a high number of these mutations. With this, we're thinking that we still need to develop a better understanding of when exactly these bacteria act. They're found pretty early, and these signatures are found pretty early in supposedly healthy cribs. So we think this may be an early life event, and right now we're trying to figure this out to much earlier detect when these bacteria cause mutations and potentially think about ways to prevent them, like by removing the bacteria in a targeted way or interfering with the action of the toxins before these mutations occur. And finally, there's a probiotic strain, E. coli nissl, which contains E. coli with the PKS operon. And we're right now checking if these bacteria could also cause these mutations. It's really early days. This is under investigation, and just finding a signature in organoids, of course, will not mean that these probiotics cause mutations in patients. And still, we think it's worthwhile looking into this and understanding if nissl E. coli can also cause mutations in patients. In my last two slides, I want to briefly present two other projects which also show how we can use organoids to rapidly understand new areas of biology. The first was led by three other PhD students, this time Mart Lammers, Jo Böhmer, and Jelte van der Vaart, and I was helping a bit with the bioinformatics part in this project. And this is about understanding how the new coronavirus infects cells, and in particular intestinal cells. Of course, we can grow organoids from many different tissues, but in the intestine, they're really well characterized. So we thought about trying this if the coronavirus can infect intestinal cells using organoids. It was based on a hypothesis which Joop observed that we have high levels of ACE2, so an entry receptor for the virus in human enterocytes. And when he and others exposed intestinal organoids to the virus, it was not only infecting the enterocytes, but also large numbers of progenitor cells, showing that it's really infecting a variety of cells in the intestine. Using transmission EM, we could also show that viral particles are indeed being formed by these cells, and finally, using bulk RNA sequencing, we could characterize the inflammatory response initiated by epithelium infected with the virus, which has also been done by now in a lot of different lung organoid lines, really trying to get a better understanding of the immediate response to this virus once it infects a cell. And all of this was happening really within a couple of months, showing that in vitro models which have a proper cell type representation can be really useful to rapidly gain an understanding of a new pathogen. And as a last project, I want to briefly talk about a human enteroendocrine cell atlas, which we built together with, again, Joep Böhmer, and also Julia Barza-Martinez and Adriana Martinez Delgado. It was published in Cell last June, and here we derived organoids from many different regions of the gut to study a rare hormone-producing cell type, the so-called enteroendocrine cell, labeling these enteroendocrine cells with genetic knock-ins for their hormones, so to be able to track the subpopulations making different hormones in real time. Again, characterizing similar to the SNAKE project with deep single-cell RNA sequencing of a number of cells, and also bulk RNA sequencing and secretomics, so characterizing the secreted products of an organoid, similar to what has been on the SNAKE project, but here in much greater detail to look for new secreted products and how hormones are being processed. And finally, and this is the concept I want to close with, organs are a really versatile model to do this discovery project, which I've been talking about a lot today, but also to do direct validation, for example, for drug targets, for any patterns which we observe in our single-cell sequencing atlas, to do, for example, receptor knockouts on these organoids, to expose them to compounds, and to be able to do rapid comparisons between human organoids and mouse organoids, to pay particular attention to interspecies differences. And as a last example here, we identified a receptor which is expressed in a subpopulation of enteroendocrine cells, and with a genetic calcium sensor, we can expose them to the known ligand and track the response of human enteroendocrine cells in real time, just serving as another example of the versatility of organoids to characterize these kinds of processes. And with that, I want to thank a couple of people. First of all, Hans Klevers, who built a really large and successful lab over the last decades and keeps pushing out great organoid stories, so really having a great time working in this lab and with everyone in there, making such a collaborative environment to build these kind of projects. Then there's Mimetas, a biotech company for building organs on a chip in Leiden in the Netherlands, and especially Dorota Kurek. I want to thank everyone involved in the SNAKE project, again, Joep and Jorik, and a large group of collaborators, mostly SNAKE people. Then the E. coli project team, again, a large international consortium. And finally, the enteroendocrine cell project team. And yeah, thanks to all of you for staying tuned for half an hour, and I hope we find some platform to discuss any of these projects. If you're interested, get in touch, and always happy to have a chat.